In the previous video I talked about the basics of how to draw genetic diagrams. In this one we're going to look at the use of genetic diagrams in working out the likelihood of inheriting a particular condition. Now there's two types of condition that we can look at. The most common, one of the common examples anyway, is of inheriting cystic fibrosis. And cystic fibrosis is caused by a faulty recessive allele. And we represent that usually with a small letter C. Now this becomes a bit of an issue because when you draw a C, if you're in a rush, it might not look like a tiny one. So you have to say that your dominant allele is a really exaggerated big C, and your recessive allele is a really small c. And that one means that you have the condition CF, cystic fibrosis, and the dominant allele is for a non-sufferer, so we're going to say normal, the normal state. Let's say the thing about having a particular condition that is caused by recessive allele means that you can have a carrier. So this means that the possible genotypes would be, we could have two little c's, we could have a big C and a little c, or we could have two big C's. Now if you have two little c's, that means because the condition is caused by the recessive allele, you're going to have CF, cystic fibrosis. If you have a big C and a little c, representing the recessive and the dominant allele, well then you're not going to have any of the symptoms, but you are going to be a carrier. It means that there's a potential to pass on that allele to the next generation, but you yourself will not experience any of the condition in the phenotype. And the big C, well here we've just got the normal dominant homozygous condition. Let's give an example. So let's say that we've got let's say parental phenotype. Parental phenotype. Shall we have let's have two carriers. So we'll have a carrier and a carrier. Parental genotype. What would the genotypes be? Well, we look at the carrier and we know that that has to be the heterozygous condition there. Big C, little c. Big C, little c. Because we've got a problem with these letters being very similar, make sure you really exaggerate them. What comes next? Well, we have to do the gametes. Remember, the gametes is separating out our two alleles and drawing a circle around them. Make them big, make them small. Make that one a bit bigger, so that one was a bit small. Make that really tiny, so it's really obvious. And then pop those into a Punnett square. Remember, this is just our grid. Much easier than drawing all those lines, getting confused. If you remember from the last video that we have the gametes on the outside, I'm going to label them to remind me. And here. I'm going to use these gametes on this side and put them down on the vertical line. Big C, little c. I'm going to use these gametes here and I'm going to put them all on the top. Big C, little c. In this stage, it's really easy to get lazy and to not write a big one and a little one and make them really obviously different. Let's have a look at what inside. Remember that inside here we've got our offspring genotypes. Got two big ones. Big one and a little one. Big one and a little one. And two little ones. Let's write those out. Offspring genotypes. If you write them out separately, we've got two big, a big and a small, a big and a small, and the littles. And here we can see that we've got our ratio. I'm running out of room here a little bit, but there's our ratio. Capital, capital, capital. These are all going to be normal. You do have two carriers, so you have to be careful. If the question asks how many are going to be carriers, you have to then look and you can say that half of them, so that's 2 out of the 4, so that would be half. If it asks you how many of the offspring are likely to, the probability of them having cystic fibrosis, then you would say 1 out of the 4, so 1 in 4 chance. If we're just looking at the condition itself, at the phenotype, well, I didn't write the phenotypes in there, but we get the idea that we're going to have normal here, and we're going to have CF, cystic fibrosis sufferer, and we're going to have a ratio of 3 to 1. Remember, you have to clarify what that ratio is. I'm going to write down that that's the phenotype to remind me. We've got 75% and 25%. Again, it may not ask you that. It might ask you how many are likely to be carriers. So that's one type of inherited genetic condition. And this specifically and importantly is how we work out if it's caused by a recessive allele. So cystic fibrosis is caused by a faulty recessive allele. And this means that you can have carriers. It's different if you have a condition that is caused by a dominant allele, and we'll look at that one next. Okay, so we need to now think about the condition, an example of a condition that is caused by a dominant allele, a faulty dominant allele. And one example is something called Huntington's disease. Now this is a quite unusual disease, and it's a neurological disease, so it's affecting the nervous system, it affects the brain, it can give you difficulty movement problems, and it also brings on dementia. So it's actually a really terrible disease, and people do tend to die quite young, well by the time they're about 40. So you might think, well, how can that be something that can be passed on? And this is because generally you don't get the symptoms until you are perhaps in your 30s, and by which time you may have already had children, especially if you don't know that you already have this condition because you haven't had any symptoms. So this is an important one to understand because it's caused by a faulty dominant allele, rather than the example that we had before, which was cystic fibrosis, and that's caused by a faulty recessive allele, which is much more common when we look at genetic disorders and their inheritance. This means that if we're going to assign some letters, let's assign the letter H. And this means that the dominant allele, capital H, and that means that you have the condition. And if you have the recessive allele, little h, and then that means you don't have the condition. So we're going to write normal. It's a normal condition for that particular protein that causes the problem. We have a look at an example of a cross. So let's have a look at parental phenotypes. Let's have somebody who is normal, non-sufferer, and a sufferer. Now let's have a think then. Parent genotypes. What genotype would somebody have if they did not have the condition? Have a think. What do you reckon it might be? Well, it can only be one thing, because The recessive allele is normal. The dominant allele means that you have it, so normal must be little h. Okay, so a sufferer. Potentially, the genotypes for a sufferer could be what? What do you think? Yeah? We could have a big H and a little h, or we could have two big H's. Either of those would mean that you're a sufferer. This means you only need one allele in order to have the condition. That is different to cystic fibrosis, which is caused by the recessive allele, where you have to have two of the faulty alleles to have the condition. I'm going to make this cross heterozygous for the sufferer, and that means we're going to do this one here, the heterozygous genotype. Let me look at the gametes then. Well, for this person, we've got two gametes that are the same, so I'm only going to write one and draw a circle around it. For this person, we've got two different gametes being formed. Draw a circle around it so we separate them so that we know they're gametes. I'm going to draw my Punnett square. Just a grid, as usual, nice and easy. Saves all the spaghetti, drawing all the lines. Remember what goes in here? The gametes. I'm going to label them. Good practice. I'm going to label these ones as well. I'm going to put this side, gametes, into here. I've only got one. I didn't need to draw this, but I did, so it doesn't matter. And I'm going to put these two gametes over the top there. We have a look next at the offspring genotypes. You've got one row to do. I've got a capital and a small, and I've got a small and a small. I'm going to write out the offspring genotypes. Remember, genotype is just the combination of the two alleles that you have. And here we have big H, little h, and two little h's. We can say what the offspring phenotypes are. Now remember, we cannot have a carrier. If you have even just one of the faulty alleles of Huntington's, because it is caused by a dominant allele, you have the condition. So here we have a sufferer. And this one does not have it, so we're going to just call it normal. We have a ratio then, potential, the probability of the offspring, of a 1 to 1. So there is a 50% chance of somebody who does not have the condition and somebody who is heterozygous for the condition. There's a 50% chance of their offspring having the condition. That actually seems quite high, doesn't it? So that's the difference compared to the previous piece of writing that I did. Remember, is that Huntington's disease is an example of a faulty dominant allele, and it means you only have to have one of the faulty alleles to have the condition. That's this one here. And the key thing is you cannot be a carrier. In this case, when it's caused by the faulty dominant allele. In the next video, I will look at sex determination and sex-linked disorders.